The human brain is a complex organ, a network of billions of neurons in constant communication. This intricate system is also intertwined with our immune system, the body's defense against disease. New research suggests that psychedelic substances may hold the key to resetting this brain-immune communication, offering potential treatments for a range of disorders. From anxiety and depression to inflammatory conditions, the implications of these findings are far-reaching. Scientists at Harvard University have been at the forefront of this research, exploring the impact of psychedelics on brain cells and the immune system. Their findings, published in the prestigious journal, suggest that psychedelics can reshape the way these two crucial systems interact. This groundbreaking research involved both mice and human cells, offering compelling evidence for the potential of psychedelics to treat a variety of conditions. The study focused on two well-known psychedelics, psilocybin, the psychoactive compound in magic mushrooms, and MDMA, often associated with its empathogenic effects. Both substances have shown promise in treating mental health conditions in recent years, but the mechanisms behind their therapeutic benefits have remained elusive. This new research sheds light on how psychedelics might be working their magic, literally rewiring the communication pathways in our brains. The implications of this research are significant. By understanding how psychedelics influence brain-immune interactions, scientists may be able to develop targeted therapies for a wide range of conditions. Imagine a world where anxiety and depression are treated not with daily medication, but with carefully controlled doses of psychedelics, resetting the brain and promoting long-term well-being. This is the future that this research hints at, a future where the power of psychedelics is harnessed for the betterment of human health. At the heart of this groundbreaking research lies a type of brain cell called an astrocyte. Astrocytes are the most abundant cell type in the central nervous system, playing a crucial role in maintaining brain health. These star-shaped cells are responsible for a myriad of functions, from providing nutrients to neurons to regulating blood flow in the brain. Crucially, astrocytes are also involved in the brain's immune response, acting as first responders to injury or infection. While astrocytes are essential for a healthy brain, their overactivation can be detrimental. Prolonged activation of these cells can lead to chronic inflammation, a state of heightened immune response that has been linked to a variety of mental and physical health problems. Chronic inflammation in the brain has been implicated in conditions such as anxiety, depression, Alzheimer's disease, and even Parkinson's disease. This is where the connection between psychedelics and astrocytes becomes particularly interesting. The Harvard study found that both psilocybin and MDMA were able to modulate the activity of astrocytes, reducing inflammation in the brain. This suggests that psychedelics may be able to restore balance to the immune system within the brain, potentially alleviating the symptoms of these debilitating conditions. The implications of this discovery are profound. By targeting astrocyte activity, psychedelics may offer a new avenue for treating a wide range of brain disorders. This could lead to a paradigm shift in how we approach mental health, moving away from simply managing symptoms to actually addressing the underlying causes of these conditions. The interaction between neurons, the brain's communication specialists, and immune cells is a delicate dance. Too much interaction can lead to inflammation and damage, while too little can leave the brain vulnerable to infection and disease. Maintaining this delicate balance is crucial for a healthy brain, and researchers have identified a key player in this process, the epidermal growth factor receptor, or EGFR. EGFR is a protein found on the surface of cells, including astrocytes. It acts as a receptor, receiving signals from other cells and triggering a cascade of events within the cell. In the context of neuroimmune communication, EGFR plays a vital role in dampening down the immune response in the brain. When activated, EGFR helps to reduce the communication between neurons and immune cells, preventing excessive inflammation. The Harvard study found that chronic stress, a known risk factor for mental health disorders, can lead to a decrease in EGFR expression on astrocytes. This reduction in EGFR disrupts the delicate balance of neuroimmune communication, allowing inflammation to run rampant in the brain. The researchers observed this phenomenon in both mice and human brain cells, suggesting a common mechanism across species. Remarkably, the researchers discovered that psychedelics like psilocybin and MDMA could reverse this effect. Administering these substances to mice with impaired EGFR function resulted in a reduction of inflammatory cells around the brain and a decrease in fear-related behaviors. This suggests that psychedelics may be able to restore the function of EGFR, effectively putting the brakes on excessive inflammation and promoting a healthier brain environment. The findings of the Harvard study offer a compelling glimpse into the therapeutic potential of psychedelics. By modulating the activity of astrocytes and influencing EGFR expression, psychedelics appear to be capable of resetting the brain's immune response, potentially alleviating a wide range of conditions. From anxiety and depression to inflammatory disorders, the implications of this research are vast and far-reaching. The study's findings are particularly promising for individuals suffering from major depressive disorder. Analysis of gene expression data from people with major depressive disorder revealed altered EGFR signaling, similar to the changes observed in stressed mice. This suggests that impaired EGFR function may play a role in the development of depression, and that restoring this function could be key to treating the condition. While the results of this study are undoubtedly exciting, it is important to note that further research is needed. The exact mechanisms by which psychedelics influence EGFR expression and astrocyte activity remain to be fully elucidated. Additionally, more research is needed to determine the long-term effects of psychedelic use on the brain and immune system. Despite these caveats, the potential of psychedelics to revolutionize the treatment of mental and physical health conditions is undeniable. By harnessing the power of these substances, we may be on the cusp of a new era in medicine, one where the brain and immune system are no longer seen as separate entities, but as intricately connected systems that can be targeted for therapeutic benefit. As research in this field continues to unfold, the future of psychedelic therapy appears brighter than ever, offering a beacon of hope for those seeking relief from the debilitating effects of brain and immune disorders.